My dear students, how are you? Hope everybody is well. HSC exam is close to you. How are you preparing? I hope everyone is preparing very well. If you don't study before the exam, you will not be able to show good skills under any circumstances. So you should all prepare well now. My dear students, today our revision class of chapter 2. We read meiosis cell division in class 11. I believe you all remember it very well. However, we will try to remember the details of the matter anyhow. Meiosis is derived from the Greek word meios, means reduce. Boveri in 1887 first time found this type of cell division in roundworm. Then Strasburger in 1883 found it in the flowering plant. Weismann in 1887 was first to point out that chromosome number of reproductive cells becomes reduced during their formation. Later on, Farmer and Moore in 1905 named it as meiosis cell division. My dear students, you know what is meiosis. I repeat, meiosis is a process where a reproductive mother cell divides twice to produce 4 cells containing half of the amount of genetic information. These cells are our sex cells, sperm in males, eggs in females. My dear students, you know where does meiosis occur? Meiosis always occur in the reproductive mother cell of diploid organisms to produce haploid gamete and in zygote of lower organisms to produce haploid organism. In this division, the nucleus divides twice and chromosome divides once. The first nuclear division is meiosis 1 and the second nuclear division is meiosis 2. Both of this division involves 2 major steps, one karyokinesis and 2 cytokinesis. You know why meiosis is called reductional division. Meiosis is a special type of division in which the diploid number of chromosome is reduced to haploid in the daughter cells. Here chromosomes divide once while the nucleus divides twice and the 4 daughter cells are formed. For the reducing chromosome number of daughter cells to half, this division is known as reductional division. Meiosis 1 includes 5 successive stages. Stages are prophase 1, metaphase 1, anaphase 1, telophase 1 and cytokinesis 1. Prophase 1. Prophase 1 is a complex stage taking up almost 90% of the meiosis division. In this stage, chromosomes become condensed. The nuclear membrane dissolves and nucleolus disappears. In this stage, there are 5 substages. Those are leptotin, zygotin, vakitin, diplotin and diakinesis. My dear students, you know about leptotin substage. This is the first stage of prophase 1. The nuclear reticulum breaks up into individual chromosomes due to chromosomal condensation and reducing water content. The chromosome remains single-stranded and thin in this substage. Bead-like swellings appear along the length of chromosome, which are known as chromomere. Nuclear membrane and nucleolus are present. This word leptotin is taken from Greek word leptos. Leptos means thin and tin means thread. At the beginning of this stage, the long, thin chromosomes are seen scattered in the nucleus. My dear students, you know about zygotin. Zygos means pair and tin means thread. In this stage, the chromosomes comes closer and form pair. This pair formation may start from the one end and finish at the other end, or from the centromere to the two sides. The process of pair formation of homologous chromosome is called synapsis. You know what is homologous chromosome? Homologous chromosome is similar chromosome coming from father and mother, which are similar in shape and arrangement of gene. Each pair is known as bivalent. The number of bivalent will be half of the chromosomes present in a cell. Nucleolus and nuclear membrane are also visible in this stage. My dear students, synaptonemal complex is a protein structure that forms between homologous chromosomes during meiosis, and it thought to mediate synapsis and recombination during meiosis 1 in eukaryotes. It is currently thought that the synaptonemal complex functions primarily as a scaffold to allow interacting chromatids to complete their crossover activities. Then pachytin. Pachys means thick and tin means thread. In this stage, the chromosomes become shorter and thicker. Each chromosome of each bivalent is found divided lengthwise, except the centromere, into two chromatids. Therefore, there are 4 chromatids in each bivalent. Each bivalent with 4 chromatids is called tetrad. The 2 chromatids of same chromosome are known as sister chromatids, and the 2 chromatids of different chromosomes are known as non-sister chromatid. My dear students, all are knowledge-based questions. What is sister chromatid? What is non-sister chromatid? What is bivalent? What is synapsis? What is synaptonemal complex? What is homologous chromosome? You have to memorize all things. Dear students, the 2 chromatids of same chromosome are known as sister chromatid, and the 2 chromatid different chromosome known as non-sister chromatid. At the end of this stage, the 2 non-sister chromatid attached and form X-shaped structures. This X-shaped structure is known as chiasma. It's also a knowledge-based question. What is chiasma? At the point of chiasma, the 2 non-sister chromatid break and exchange their parts. The interchange of the parts of 2 non-sister chromatids are known as crossing over. As a result of crossing over, The change of characters of the chromosome are taken place, which leads the change of species. Nucleolus and nuclear membrane are also present here. Then diplotin. Diplose means double and tin means thread. In this stage, the repulsion may start many points at a time. As a result of repulsion, one loop or more than one loops are formed in between two chromatids. Gradually, chiasma move towards the end of chromatids. This movement of chiasma towards the end is known as terminalization. My dear students, you have to memorize what is terminalization. Nucleolus start to disappear here. Nuclear membrane are also seen in this stage. Then diakinesis. Dia means other side and kinesis means division or arrangement. In this stage, the chromosomes become further thickened and shorter due to deposition of matrix. Terminalization is still going on and the part exchanged chromosome separated at the end of this step. This is known as complete terminalization. Nucleolus and nuclear membrane disappear at the end of this stage. Crossing over. The exchange of some pairs or genes between the two non-sister chromatid of paired homologous chromosome is known as crossing over. In 1909, Thomas H. Morgan observed crossing over in maize plant. Okay, now mechanism of crossing over. At first, homologous chromosome comes together to form pair or synapse. The paired homologous chromosome divided lengthwise, except centromere, into two chromatids. The same chromatids of same chromosome are known as sister chromatid, and the two chromatids of different chromosomes are known as non-sister chromatid. You know, it's also a knowledge-based question. What is sister chromatid? What is non-sister chromatid? You have to memorize it properly. Then, chiasma or egg-shaped structure are formed in between non-sister chromatid. Then, at the point of chiasmata, the two non-sister chromatids breaks down. Then again, join with the other chromatids pairs. Thus, they exchange their pairs. So, the process of crossing over takes place in following stages. First one, synapse. Second one, duplication of chromosomes. Third one, chiasma formation. And last, terminalization. And then, exchange of chromatid pairs. My dear students, in this picture, this is the homologous chromosome with genes being represented by different letters of the alphabet. The genes on one chromosome are shown using capital letters and that on the homologous pair using small letters. My dear students, significance of crossing over. It produces new individuals having new combinations of traits. Crossing over has helped in establishing the concept of linear arrangement of genes. The frequency of crossing over helps in the mapping of chromosome, for example, determining the location of genes in the chromosome. Variation of character. Except identical twin, there is no species which exactly the same character of sexually reproductive organism. Variation of characters are due to interchange of pairs of chromosome by crossing over. origin of species as a result of crossing over. New species or variety is created, which leads evolution. Gamete formation and reproduction. Male and female gametes are formed through the meiosis cell division in diploid organisms. By the union of the male and female gamete, sexual reproduction is done. Okay, my dear students, difference between mitosis and meiosis. Generally, mitosis cell division occurs in somatic cell or body cell, and generally meiosis cell division occurs in reproductive mother cell, and then mitosis cell division, this process occurs both haploid and diploid cell, but in meiosis cell division occurs only in diploid cell. Nucleus divide once, but in meiosis cell division nucleus divide twice. The number of chromosome in daughter cell are same as the number of mother cell, but in meiosis cell division the number of chromosome in daughter cell become half of the mother cell. 2 daughter cells are produced, and here 4 daughter cells are produced. There is no crossing over between the chromatids, but there is crossing over in between 2 non-sister chromatid. It is known as equational division, and meiosis cell division is known as reductional division. My dear students, we have to memorize another difference between metaphase and metaphase 1. Metaphase of mitosis cell division and metaphase 1 of meiosis cell division. Metaphase cell division, at this stage, the centromere is in the equatorial line, and the arms of the chromosome remains towards the pole. In metaphase 1, in this stage, the centromere located at the pole and the arm at the equatorial line. Centromere is divided longitudinally, and at a result, chromatids are separated with each other. But in metaphase 1, centromere remain undivided. So the chromatids of the centromere remain attached by the centromere. Then we have to memorize another difference between anaphase and anaphase 1. Anaphase cell division, anaphase, this stage is, in this stage, anaphase stage, only daughter chromosomes moves towards the pole, and in anaphase 1, whole chromosomes moves towards the pole. Equal number of daughter chromosomes move towards the pole. Half number of chromosome move towards the pole. In this stage, the divided centromere remain with the daughter chromosome, and in anaphase 1, undivided centromere remain with the chromosome. What is interkinesis or interphase 2? Interkinesis is the period of rest between meiosis 1 and meiosis 2. No DNA replication occurs during interkinesis. My dear students, we already know what is chromomere. Some definitions of this meiosis part we want to review. What is chromomere? My dear students, bead-like swelling on chromosome in leptogen substage of meiosis cell division is known as chromomere. What is tetrad? My dear students, you know 4 chromatids of a bivalent is known as tetrad. Then what is homologous chromosome? Similar chromosome coming from father and mother, which are similar in shape and arrangement of genes. My dear students, what is synapse? You can say, what is synapse? The process of pairing homologous chromosome is known as synapse. What is terminalization? Movement of chiasma towards the end of the chromosome, known as terminalization. What is bivalent? Paired homologous chromosome are called bivalent. What is chiasma? X-shaped structure is known as chiasma. What is crossing over? Exchange of parts takes place within the non-sister chromatids of homologous pair during Pachytene stage, which is known as crossing over. And what is interkinesis? The resting stage in between meiosis 1 and meiosis 2. We gave you some sample questions. Hopefully you can answer all the questions if you listen very carefully that we have discussed now. And I think you can understand every questions and can easily answer all the questions. I hope you have listened carefully and remember everything. My dear students, this is a creative. We provide you this creative question for assessment 3 test. My dear students, number 1, what is interkinesis? You say, what is interkinesis? Now you say. I hope you can. Now you can. Why meiosis is called reductional division? You can. Then describe the third, second or fourth sub-stage of the Y division of the stem. What is Y division? Y division is the meiosis cell division. Second sub-stage is zygote. What is the third sub-stage of meiosis? Third sub-stage is Pachytene. What is the fourth sub-stage? Diploide. Okay, now we can. Then write down the importance of the process occurring in the third sub-stage. Third sub-stage is Pachytene. Which process occurring here? Do you understand what is wanted in this question? What process has stimulus? What process has been talking about? In Pachytene stage, the process talking about in Pachytene stage is the crossing over. So you have to write down the importance of crossing over. Then another question we provide you. This question, what is homologous chromosome? You know what is homologous chromosome. Write down the difference between mitosis and meiosis. We recently read it. Then describe the process of the stem. Describe the process of the stem. What's the process? Here we can see the exchange of chromosomes. Some part exchange here in between non-sister chromatid. So this is the crossing over, and you have to describe the process of crossing over. Then describe the importance of the division of the stem. First, the division of the stem. This is crossing over, and crossing over occurs in meiosis cell division. So you have to describe the importance of meiosis cell division. I think if you listen properly, you can answer every questions, and you can catch every questions. You can understand every questions. Read properly, listen properly. All are easy to you. Thank you all to stay with us. See you again. Bye bye.